organize and, ooh, this meeting is being recorded. It's designed to organize and help people meet people who are interested in both medicine and research. A frequent career path for physician scientists is to pursue the MD-PhD route, even though that's not necessary to become a physician scientist. You just have to have interest in research and medicine. A lot of times, a lot of people think that you can't combine those two interests. They think that they're mutually exclusive. You either do research or you do medicine. But there are a lot of people that do both. There are PhDs that do clinically relevant research, and there are MDs that do a lot of research. So we're designed to help meet people, and this is right here is a journal club, and we're reviewing some interesting papers from UW. We have a paper from Dr. Lexi Walls up right now, and she discovered some properties from the spike glycoprotein for the coronavirus that we're all aware of. And this is proven to be really helpful for vaccine development. And you know, this is just one of the papers that we're planning on reviewing, and this will like help you get acquainted with research. And we are also planning on doing some other more career development options, like interviewing, how to interview, how to take the MCAT, how to, you know, find a research lab, stuff like that. And to really branch out your options and get the most out of your undergrad experience, because UW is a big public research institute and you could really benefit here. So I went out, I kind of mumbled on a little bit about this club, but we could start now with our speakers for today. Sunny, do you want to take it away?
Yeah, okay. So I'll first welcome everybody to join us today in our second journal club. And today we're here to discuss about the structure, function, and antigenicity of the SARS-CoV-2 spike glycoprotein, which is a research paper that is published back in April 2020 by the Research Lab on UW. Yeah. And my name is Sunny. I'm one of today's presenter, and I'm currently a junior enrolled in University of Washington, majoring in biochemistry, and I have my co-presenter Peter with me.
Hi everyone, my name is Peter Chang, and I'm a junior student majoring in molecular, cellular, and developmental biology, and I'm studying in the UW as well. And I'm very happy to be here today and share with you about this research.
Yeah, thank you. And we have really, we are really honored to invite Dr. Lexi Walls from Research Lab to join us here as our guest speaker today. Hi, Dr. Walls. Oh, she's there. It's okay.
Yeah, I'm here.
Oh, hi.
Hi, everyone.
Yeah. Hi. So she's an expert in structural biochemistry and coronavirus research. Yeah, so she'll be here with us reviewing our work.
And also, like Daniel was saying, I went to grad school and I did research as an undergrad, so I'm happy to answer more general questions about that as well, if that's helpful to anyone.
Yeah, thank you. So, okay, let's get into the topic. So before we get started, I would like to give you guys an overview for coronavirus. So coronavirus is split into 4 genera, which is alpha, beta, delta, and gamma. And delta and gamma coronaviruses are majorly the coronavirus that infects birds. And as for alpha and beta coronavirus, they are mostly they are for mammals. So and for alpha CoVs, they are mostly associated with pigs, like dogs and cats, and there are also some human coronaviruses, but this is not our focus today. So what we're focusing on today is the beta coronavirus, and it consists of some more like milder human coronaviruses that causes like mild fever or like common cold symptoms like coughing, sneezing, or anything. Yeah, and there are some other more severe coronaviruses that are more well known of, such as SARS-CoV and MERS-CoV, and also the novel SARS-CoV-2. Yep. So here are the beta coronaviruses. It consists of 4 low pathogenicity endemic human coronavirus, which are listed here, which I said causes like common cold symptoms. And here it also consists of some highly pathogenic epidemics and pandemics. And those 3 coronaviruses we are pretty familiar of. So why is knowing the structure of the spike protein so important?
Yeah, so as a recap from our last journal club meeting, we learned about how the coronaviruses are able to fuse with the host cell. So to talk a little bit in detail here, let's look at these purple triangles here. These purple triangles are called the transmembrane spike glycoprotein. And usually we just call it spike protein and abbreviate as S. And these spike proteins, when they enter the body, they will look for this receptor, this enzyme called ACE2. And what this ACE2 typically does in the human body is that it regulates blood pressure and volume. But in this case, the SARS-CoV virus is present, and the spike proteins will take advantage of this enzyme and use it as a receptor into the host cell. So after the binding of the spike protein and the ACE2, it will promote the cell entry, virus entry into the host cell, and further the replication process. And because of the spike protein is the most exposed and known to directly interact with human cells, so it's important for us to determine its structure. And how we're able to do it is we use this method called the cryo-EM. And what this is, it's called the cryogenic electron microscopy. And this microscope is special because it uses electron beams instead of light beams. And because the wavelength of the electron is much shorter than light, even so it can produce even finer details than what light microscopes can produce. And so this cryo-EM compared to traditional electron microscopes. The traditional ones, they often, the electron beams are so high in energy and heat, it often just destroys the sample that we want to look at. So in this case, in cryo-EM, we are able to freeze the protein samples that we want to look at. So it does not get destroyed by the electron beams. So if we look at here, the electron beams shine onto the frozen spike protein samples here. And these spike protein samples are all in different orientations. So the deflections of the the structures are able to be caught on these electron detector. And then the projected images are able to be sorted into different orientations. After these are sorted out, the computer is able to combine all of these 2D pictures and make it into a 3D structure, like this one on the left here. So it's a very crisp and clear image that we can use.
Talking of the structure of Polyspike proteins. And Polyspike proteins are composed of two subunits, which is S1 subunit and S2 subunit. And S1 subunit is the outer part of the spike protein that is where the virus is directly exposed to the surrounding environment, and it is comprised of the receptor binding domains, and it also serves the function of stabilizing the prefusion state of S2 subunit. And as for the S2 subunit, it contains the fusion machinery, and it is covered under the S1 subunit. And it is also the subunit that is protruding out of the virus. So here is the monomer, like a single unit of the that composed of the spike protein, and we can see that colored in the darker blue and the cyan blue, which stands for S1 subunit and S2 subunit respectively. And here colored in red is the S1 and S2 subunit cleavage site. So it was initially a bulky protein, and then it would be cleaved at some point to make it into subunit 1 and subunit 2. And there's also another cleavage site that is important for the spike-mediated entry into cells, is the S2' cleavage site. So its function is to activate the protein for membrane fusion, which we talked about in previous slides, saying that S2 subunit contains the fusion mechanism. So the S2' cleavage site is what it's talking about. As for the overall structure of the spike protein, so what is shown in previous slide is the monomer of the spike protein, and the actual spike protein is composed of 3 of those monomers, making it a trimer. So for the diagram labeled B, it is a side view of the spike protein, and the diagram labeled C is the top view of it. And so here, to make it simpler to explain the next slide, I would like to replace it with my personal drawing of it. And the colors are corresponding to each of the monomers. And those labeled A and B are the different binding domains of the spike protein. So here is an overview of how like the spike protein mediates the viral entry. So for these two on the top left, those are those milder CoVs. So what they use is the domain A receptor binding domain, yeah, to bind to the its receptor, which is written down here, the chemical here is their receptor, and that result in viral entry. And for MERS-CoV, it uses the domain A to recognize the non-acetylated sialocyte, and that promotes the domain B to bind to dipeptidyl peptidase 4. And the last one, dipeptidyl peptidase 4, is its actual receptor, and that result in viral entry. And for SARS-CoV and SARS-related CoV, they use domain B to directly interact with ACE2 receptor, which Peter talked about in previous slides. So what is interesting is that SARS-CoV-2 uses ACE2 to enter the cells as well. So how is this determined? So an experimental method that they use is the murine leukemia virus pseudotyping system. So murine leukemia virus, abbreviated as MLV, is a kind of virus that will infect murine. So murine is this cute experimental mouse here on the slide. And yeah, it causes the mouse cancer. And so what exactly is pseudotyping? So here we have two viruses here. One is spherical and another is like bullet shaped. So what we're interested in is to test how the spike protein of the spherical virus is effective in its spike mediated viral entry into its host cell. But so what we do is to put this spike onto another virus, which might be easier for us to control, and we can use it on murines. Yeah, so it's basically just make this new virus that is called, which is a pseudovirus, that can help us test the pathogenicity that is mediated by this spike protein. So what they do is they put two so what they do is they put SARS-CoV-2 spike-bearing pseudovirus and SARS-CoV spike-bearing pseudovirus together, and they make them attack the viral E6 cells. And it is a cell, kidney cell of African green monkey, and it expresses ACE2 receptor. So what results in is that the viral entry went equally well. So what can be concluded about that is that ACE2 of African green monkey is a functional receptor for SARS-CoV-2. And because this is pretty helpful, because monkey is like closely related to human, but it is not too ideal because monkey is not human, and its ACE2 receptor is not human's ACE2 receptor. So what they do next is that they take the SARS-CoV-2 spike-bearing pseudovirus to attack baby hamster kidney cell, and which is transient transfected with human ACE2. So what is transient transfected? It is, so with some method, they put human genome containing that would make the cell express humans ACE2. They put that section of genome into baby hamster's kidney cell, and then that baby hamster kidney cell would then express human ACE2 receptor on its cell membrane. But this effect is temporary because our genome is not incorporated inside their own genome, so it wouldn't persist forever. But it would definitely has a period of time of effect on it. So when that is still effective, while the cells still have human ACE2 receptor, they find that the baby hamster kidney cells become susceptible to transduction with SARS-CoV-2. And then you can conclude that the human ACE2 is a functional receptor for SARS-CoV-2. So how is SARS-CoV-2 different from other coronaviruses? So for the amino sequence that is determined for SARS-CoV-2, they found that there is a unique insertion of furin cleavage site, so which is here, yeah. And so for the related SARS-related coronaviruses and SARS coronaviruses, they all doesn't contain this insertion of protein sequence. So this insertion is definitely unique to SARS-CoV-2. And so they want to see if this novel change in structure would have any effect in its spike-mediated viral entry into host cells. So what they do is that they take out the furin recognition sequence and they substitute it with I and L, the two amino residues. So what are they trying to do is that they get rid of furin recognition sequence. So furin cleavage wouldn't happen because there's no furin cleavage site anymore. And two, they mimic the SARS-CoV-S and SARS-related CoV-S subunit 1 and 2 cleavage site, so which is this IL residues. So what is the difference between the two cleavage sites? So for the SARS-CoV-2 furin cleavage site, cleavage, it happens during biosynthesis. It's biosynthesis in Golgi apparatus of the host cell. So it happens inside the host cell. While the SARS-CoV spike subunit 1 and 2 cleavage happens upon interaction with the target cell. So one happens inside the cell and one happens outside of the cell. So here is a Western blot result for the 3 viruses. One is one is SARS-CoV, one is SARS-CoV-2, and one is SARS-CoV-2 mutant form, which is without furin cleavage site. Okay, so what Western blot do is that it detects protein based on its molecular weight. So here on the left, we can see that it has a bunch of numbers, which indicates their molecular weight. And for SARS-CoV spike and SARS-CoV-2 transform spike, they have similar molecular weight, because we know that the cleavage happens when they're in contact with their host cell. So right now, as it is just a spike protein, so it is not cleaved yet, so it has bigger molecular weight comparing to SARS-CoV-2 spike, which its cleavage happened during its biosynthesis process. And here are the results of the experiment. They use the three the pseudoviruses bearing those three different proteins, spike proteins. So here for the viral E6 cells, which is the African green monkey kidney cells, they observe a pretty similar pseudovirus entry of SARS-CoV-2 and SARS-CoV pseudovirus into the host cell. But what is really weird is that for the SARS-CoV-2 mutated form, it has a significant increase in the pseudovirus entry. But for the baby hamster kidney cells, it observes the opposite trend. So let's talk about the control first. Here, the first two category of this data has the baby hamster kidney cells with just the human ACE2 receptor. And the next one, it has just SARS-CoV-2 spike pseudovirus, but without human ACE2 receptor. So we see that if there's only one present, either the virus spike in the or the human ACE2 receptor, there wouldn't be any very low pseudovirus entry into the host cell. As for the comparison for SARS-CoV-2 spike and SARS-CoV-2 mutant spike, it has an opposite trend comparing to viral E6 cells. So what we can conclude for the experiment result is that first one, whether the cleavage happened during biosynthesis, which is inside the host cell or outside the cell, is not necessary an effect for spike mediated entry, because we can still see that the mutant form and the original form of SARS-CoV-2 still has their pseudovirus entry. So they're still functioning. And the second conclusion we can draw is that the polybasic cleavage site, which is the furin cleavage site, I think I forgot to talk about it, but it consists of several arginines, amino residues. So an arginine is a basic residue. So it's called polybasic cleavage site. And it seems to enhance SARS-CoV-2 transmissibility for human ACE2 receptor.
Right. So now we know that the spike proteins, the SARS-CoV, both the SARS-CoV-2 and SARS-CoV spike proteins, they bind to human ACE2. Now we want to compare them. So in this slide, we want to look at the human ACE2 affinity to SARS-CoV-2 spike and SARS-CoV spike. And if we look at the bottom graph here, this is the amino acid sequence for the SB domain, which is the domain that binds with the human ACE2 receptor. And though the sequences are highlighted in red, represents that they have the same sequence for SARS-CoV-2 and SARS-CoV. And the ones that are in white means that they are different. And if we do the math and count do the math, and then we can find that SARS-CoV-2 and SARS-CoV shares about 75% of overall amino acid sequences for their SB domain. And there's also these asterisks below some of the amino acid residues. And these asterisks are the receptor binding motifs. And what these motifs are is that they are some of the key residues that helps the spike protein to bind with ACE2. So if we look at these important sites, motifs, residues, that we can find that about 50% are identical between SARS-CoV-2 and SARS-CoV. And now if we have our attention to the top right here, the two graphs here. The top graph is representing the binding response of SARS-CoV-2, and the bottom graph is representing the binding response of SARS-CoV. And by just looking at the graph, we can see that they have pretty similar trend of binding. Their shapes are very similar. But if we look at the actual binding response, the highest, the red line here, it's around 0.3 for SARS-CoV, but the binding response for SARS-CoV-2 can achieve almost at 1. So we want to look further into this, and so we want to look at the KD of both the SARS-CoV-2 and SARS-CoV. And what KD is the dissociation constant. And a higher dissociation constant means that fewer spike proteins are bound to the ACE2 receptor. And lower KD dissociation constant means that fewer spike proteins are not bound to ACE2. So what we can conclude from that is lower dissociation constant equals to higher binding affinity of spike and ACE2. And if we look at the numbers here, we can see that SARS-CoV-2 spike protein has almost 4 times the affinity of compared to SARS-CoV spike. Although this looks like a significant increase in the binding affinity, but it's still only a 4-fold increase. And if it's under a 10-fold increase, it's hard to say if it's significant or not. So we need more evidence and to see if SARS-CoV-2 really has a higher affinity to human ACE2 receptors. And now we want to look at the structures of SARS-CoV-2 spike glycoprotein in 2 conformations. And the 2 conformations are or one of the confirmation is that one of the SB domains is open, and the other one is that all of the SB domains are closed. So if we look at the structures of both the SARS-CoV and the MERS-CoV, we are able to see observe that only one of the SB domains is open. So it looks like this. And if we observe if we look at the 4 other human CoVs that has a lower pathogenicity that causes a mild fever accord, they have all of their SB domains closed. So this leads us to think to the discovery of that the SB domains are open to expose the receptor binding motifs, which means that the spike proteins bind to the human ACE2 receptors better. So now we can conclude that an open SB domain is linked to a higher pathogenicity, and a lower and a closed SB domain leads to a lower pathogenicity.
Okay, so Peter just talked about the 2 confirmations of the spike protein and its possible connection with the pathogenicity of SARS-CoV-2 virus. Now we're here to talk about the antigenicity of SARS-CoV-2. So it's basically answering this question. So what's on the slide? Yeah, how's SARS-CoV-2 spike-mediated entry and host cells respond to antibodies? So here is another important structure that is detected by the cryo-EM, and it's called the heterogeneous end-link glycan. And what that is that is an oligosaccharides. They are oligosaccharides that are attached to the proteins. So oligosaccharides are basically sugars that are composed of 3 to 10 sugar monomers. Yeah, so it's just little sugar balls that is distributed all over this spike protein, and we can see on in the diagram that it is colored in dark blue, and yeah, it's like all over the surface of the spike protein. So it has several functions. According to the paper, it says that it participates in the spike protein folding, and it affects the priming by host protease, and it might modulate antibody recognition. So to sum up its function, it's basically end-link glycans are pretty important to the structure maintaining of the whole spike protein. Oh, and yeah, can you hear me? Okay. Oh no, I'm not done. Sorry. Sorry. So when, yeah, so the end-link glycans are pretty important for maintaining structure of the spike protein, and as well as it might be some sort in charge of the spike protein interaction with the antibody. So what they discover is that the SARS-CoV spikes and the SARS-CoV-2 spikes, they have pretty similar sites of like the N for end-link glycan, for the glycan to bind to the protein, and their condition of binding the glycan is pretty similar. So what we can expect from this observation is that SARS-CoV and SARS-CoV-2 might have a similar response to antibodies.
All right, so other than the N-linked glycan, we drew out the amino acid sequences for both the SARS-CoV spike protein and the SARS-CoV-2 spike protein. And what we found that the S2 fusion machinery is a lot more conserved in the S1 subunit in both the SA and SB domain. What that means is this S1 subunit is very varied between the SARS-CoV and SARS-CoV-2. And so we want some observations to back up our discovery. So what we know is that not all viruses use ACE2 as entry receptor. And what that means is because the S1 subunit and the SB domains are in charge of binding to the ACE2 receptors. So if not all viruses use ACE2 as entry receptor, they will have varying amino acid sequences for their S1 subunit. And also another observation is that more exposed parts are likely to be subject of a more strict selection pressure from our immune system. So based on the observations we have above, we can kind of conclude that the antibodies, they target the highly conserved S2 subunits. It doesn't necessarily mean that the antibodies bind to S2 better. It's just because that the S2 subunits are very conserved across the SARS-CoV spike and SARS-CoV-2 spike. So the antibodies are able to find the same sequences to bind. So this is like the grand finale of the whole research. So in this experiment, we immunized these mice with the SARS-CoV spike proteins. So when these mice are immunized, they are able to produce these antibodies for SARS-CoV, and these antibodies are able to inhibit the activities of SARS-CoV spike protein. If we look at the top picture here, we can see that the SARS-CoV spike and SARS-CoV-2 spike are present. And they are able to, when the antibodies are not present, they are able to bind with the ACE2 receptor equally as well. And there's cell viral entry into this host cell are equally as well too. But if we look at the bottom picture here, when these antibodies for SARS-CoV are present, you can see in this diagram that the SARS-CoV-2 spike protein actually gets inhibited by the SARS-CoV antibodies. So it doesn't bind to ACE2 as well as compared to when there's no antibodies present. And there's a significant reduce in the whole cell entry as well. So we can now look at a more detailed bar chart here. So if we look at SARS-CoV spike and SARS-CoV-2 spike, when there are no antibodies present, when there are no SARS-CoV antibodies present, they have equally as high of a viral entry percentage, almost 100%. But when a SARS-CoV antibody is present, we can see that the SARS-CoV viral entry percentage is near to zero. There's nearly no viral entry in this case for SARS-CoV. And for the case of SARS-CoV-2, we can see that using the antibodies of SARS-CoV, nearly a 90% decrease in viral entry is observed. So this is very interesting, very exciting. But we need to know that it's not a perfect inhibition, but it's very close. Right. So conclusions for our presentation today is that this is a great intro to structural immunobiology and is used to help with vaccine development. So we listed 5 points that help us recap. So the first is that the SARS-CoV-2 uses ACE2 to enter target cells. And the second point is that the SARS-CoV-2 has a unique furin cleavage site, and it has possible positive effect on SARS-CoV-2 entry into host cell. And number 3 is that SARS-CoV-2 and SARS-CoV bind with comparable affinities to ACE2. Although SARS-CoV-2 shows a almost 4 times better affinity to compared to SARS-CoV, but we still need more information and more researches on how if it's significant or not. And the fourth point is that the structure of SARS-CoV-2 spike glycoprotein has 2 conformations. One is open and one is closed. And the open conformation relates to a higher pathogenicity of the virus. And a closed SB domain relates to a lower pathogenicity virus. And the last point is that mirroring SARS-CoV polyclonal antibodies significantly decreases SARS-CoV-2 spike-mediated entry into cells to about 10%. Right, so that concludes our presentation this time, and we're accepting any questions. And hopefully Dr. Walls can help us answer these questions.
I had a question. So I'm trying to think of a way to word this, but this might be like over like bigger than this piece of research alone. But what if the virus mutates and we can like can we repeat this sort of experiment and like figure out find other antibodies that might be able to fight a different strain of COVID? Or do you think that there would be like there would need to be a different kind of approach?
Do you want me to answer?
Yeah, I guess this is for you.
Sure.
These questions.
Sure. I think it's a great question. So the whole virus is mutating, but it's mutating much slower than most other viruses that we hear about. So like compared to flu, it's essentially not mutating if you compare it to flu. That being said, there are a couple known mutations specifically in the spike protein, and people are constantly looking at how those are affecting things like transmissibility, entry, the ability of antibodies to neutralize. So people are looking at this, and in general, the single mutations that we're finding in spikes, in general, if you've had COVID, you will have antibodies that are targeting more than just one region on the spike. So even if one of your antibodies, let's say that mutation prevents that antibody from working, in general, you have another one, or that can come from a different angle. So in general, most people who have naturally had COVID or who will be vaccinated will be able to handle mutations that will arise.
Thank you. And also kind of like a follow-up question to that. Is there like a particular reason why it's mutating more slowly? Like, is it trading off like how viral it is for mutability, or yeah, I'm not sure.
There's a lot of reasons why certain viruses mutate faster than others, and a lot of it has to do with how the virus replicates. So how good the virus is at replicating itself, and also how likely it is for there to be large genome rearrangements. So flu is very common for having large genome rearrangements. Coronaviruses in general have a lot more checkpoint mechanisms to basically prevent that from happening. Coronaviruses also have one of the biggest RNA viral genomes, and because it's so big, it needs to basically have a way to maintain itself. It's kind of like, you know, humans have a huge genome, and we have a lot of ways to make sure that that genome doesn't get messed up. Viruses have significantly fewer ways to do that, but the bigger they are, in general, the less mutations they'll have, especially with coronaviruses.
Okay, cool. Thank you.
Yeah, great question.
Okay, we would like to insert one of our slide questions here. Yeah, so what is the protein homology of receptor ACE2s in different species?
Yeah, so a lot of people have started looking at this, and in general, most animals and most animal cell types are SARS-CoV-2 is able to bind their ACE2. So the homology is really high. I don't have a specific number off the top of my head, but yeah, that's why, you know, you're hearing about cats and dogs and minks and pangolins and all of these things. It's because the like lung or nasal passages of all of these animals have the necessary receptor, so ACE2, and also have the necessary proteases. So you saw in the presentation that the spike needs to be cleaved in order to enter. So if you have those two things, the virus will be able to infect.
Thank you. Any more questions?
I actually have a question for Dr. Walls. What is it like being on kind of the forefront of some really kind of, I don't know, to me it's like life-changing kind of what you're doing, and I think it's just it's unbelievable. I was wondering if you could kind of speak to what that's like.
Yeah, I guess I can tell you some vignettes. So on January 10th, that's when the sequence of SARS-CoV-2 was released. And basically that's the day that we kind of started sprinting. So as soon as the sequence was released, you know, we're trying to figure out where's the spike protein, what does the spike protein look like? Because at this point it wasn't called SARS-CoV-2, right? It was called novel coronavirus. No one had any idea what it was going to be related to, how similar it would be to other things. And so we initially, you know, we already started thinking like, what experiments can we do? How can we figure out what's happening? And so that was kind of a crazy, mad rush. And this was really before the pandemic hit the US. So, you know, I was working like crazy and regular life was still going on outside, but, you know, slowly things started shutting down or fewer people coming into work. And it's been very strange, very humbling. But it's also, if I'm allowed to say, it's been fun because, you know, we're learning so much at such a fast rate, and there's a lot of people are interested in the work. So I've had lots of good conversations with scientists and non-scientists alike, just trying to, you know, understand what is ripping through our society. So it's been a lot of things.
Awesome, thank you very much.
Yeah, and you know, this was kind of our first story, but we just recently, we have a vaccine candidate. We've looked at a bunch of antibodies that are in clinical trials. Like, this is really the first time that, you know, our style of research is helping people. So that's also crazy and cool and exciting.
I have a question.
Oh.
It was on like the relative speed on how this is all being discovered. So everyone's heard that like we've sped up the vaccine process by like a ton, from years down to a few months. And I was wondering how your pacing in lab changed.
Yeah.
Once that sequence was released. Was it just crazy after then?
Yeah, I don't think you want to know the answer to that. Yeah, for this paper, we were working pretty much round the clock. So yeah, it was a lot of hard work, but it was also, you know, there was strong motivation behind it. So, but yeah, it's been also a journey, and you know, as everyone's had during 2020 of, you know, setting your own boundaries and, you know, how much can you work, when does working more not actually help anything? So everyone's human too, right?
So I had a question about how maybe you would compare this virus with like another virus, maybe like maybe not something like the flu, but like actually yeah, maybe the flu is like a good example, but just like how, like what makes COVID so much more lethal? Is it because of its ability to bind to ACE2, or yeah, it's kind of like the question, like, is it because it's more complex, or maybe like bigger, or yeah?
I think if I or anyone knew the answer to that, we would all not be at home right now, and we would be probably in a lecture hall. I don't think anyone has any idea. One of the main, like, this is not a reason, but one of the main things that people are noticing, you know, is it spreads potentially easier than flu. So that's why so many people are getting it. And then there's such a wide range of symptoms and severities that isn't really seen with, at least we don't know if it's seen with flu. We haven't seen things like this before.
Yeah.
And that's mostly due to how the immune system is interacting with SARS-CoV-2. And yeah, I wish I had an answer. But it's definitely not because of ACE2. So one of the common colds caused by coronavirus, NL63, binds ACE2. So
okay.
Yeah.
So it's definitely not because of the receptor then.
No. Or at least that's not the whole story, if it is part of the story.
Right. All right, thank you.
Yeah, no problem.
I have a question.
Yeah.
So, has anyone ever tried using like human SARS-CoV antibody to like deal with this COVID-19?
Yes. So all of the antibody, the monoclonal antibodies that are in clinical trials are either isolated from people who have been infected either with SARS-CoV-1 or SARS-CoV-2, and they've been isolated, and then, you know, we've learned their sequence, and then we make them out in the lab. So absolutely, those antibodies are being used. And also, you may have heard of convalescent therapy. So what you can do is, if you've had COVID, you can donate blood, and then people can basically isolate antibodies from that blood and use that as therapy for COVID. It hasn't been shown to be super successful yet, but it does tend to shorten people's times in hospitals and maybe some severity, but it's still early to say.
So, if those antibodies can be like isolated from human blood, is it possible if we can like determine the structure of those antibody and make some man-made antibodies?
Yes.
Is it? Oh, okay.
Yes. So people are doing this. So we've solved structures of some antibodies, but a lot of groups all over the world have solved structures of antibodies. And then just for an example at UW, the Baker Lab they've designed not antibodies, but they've designed inhibitory binders. So basically they took the structure of SARS-CoV-2 spike that we solved. They took the structure of ACE2 and they said, can we design something to prevent those two things from coming together? And that's also in clinical trials right now. So yeah, totally a valid way of doing things, and a lot of people are trying it. Great question.
Thank you. So, yeah, if you mind going to the next slide.
Well, I actually have another question.
Okay.
So the spike proteins bind to human ACE2, right? But human ACE2 originally have other functions, such as they control the blood or something. But if there is if someone is infected by this by this virus, does the original function of the ACE2 protein do they still have that function, or is decreased?
I don't know. I don't think it's decreased, but I'm definitely not the expert on human ACE2 biology. But yeah, ACE2 absolutely has a role, and the virus has been shown to adjust the level of ACE2 at certain cell types, but I don't think we yet have a good picture of what that's doing or why that's a big problem. But it's tantalizing to think about, you know, ACE2 stands for angiotensin converting enzyme. And so, you know, there are a lot of heart issues associated with COVID. And so it could be related, but I don't think we know yet, but it's a great question.
Yeah, thank you.
Yeah, this is a thing that I'm really interested about. Like, so I see this receptor in the paper as well, but it's not like covered much.
Yeah, so TMPRSS2, it's not actually a receptor, it's a protease. And so this is at the cell surface, the same region that ACE2 would be. And it's thought that this, during native infection, is required to cleave that S2 prime cleavage site, which basically primes the spike for membrane fusion and for eventual infectivity. And so it seems like the combination of these two things is really important. Just having ACE2, the spike can bind and the virus can sit at the membrane, but if there's no cleavage, it's going to be really inefficient at actually entering into the cell. So it needs kind of both of these things. It needs a protease, and it also needs a receptor. And that kind of relates to whether or not the virus can enter a certain cell type.
Mm-hmm. So sort of like a follow-up to this. So it seems like two of them are, this question. So is there any vaccine research that targets TMPRSS2 instead of ACE2?
Yeah, so the three vaccines that are farthest in clinical trials are all based on the entire spike protein. And so what that means is that your body can create antibodies against any portion of the spike. So that includes the ACE2 binding site, that includes the cleavage sites, and that includes other sites that are may or may not be supportive of blocking infection, and more of that is coming out now. As far as targeting human protein, so like one nice thing to think about is like, oh, if we just block ACE2, then the virus can't enter. But like we talked about earlier, ACE2 has a role that's really important for the body. And same thing, if we just give people, you know, TMPRSS2 blockers, sure, we might have less SARS-CoV-2 entry, but we also are messing up the function of this protein that's critical for our current homeostasis. So it's a tough balance.
Sounds. Okay. So,
go ahead.
Sorry. So I'm thinking, like, so recently, what has gone the furthest is still like targeting ACE2. I mean the vaccine design, ACE2 binding site.
Yeah, I mean the mRNA vaccines that are farthest in clinical trials, I mean, a lot of the antibodies that people make against, like because of the vaccine, are probably blocking ACE2 binding, but there's probably a lot of other antibodies created by, like, once you've taken the vaccine that bind to other regions as well.
Mm-hmm. Thank you. All right, actually, I think we can proceed to the last question on our slide. So it's just, what research are you working on right now? Any updates on this research?
Yeah, so I think the biggest thing that we're working on right now is we have antibodies and vaccines in clinical trials. And so we're just trying to push those forward as best as we can. So antibodies, kind of like we talked about earlier, would be given once you have an infection, and typically they'd be given, you know, if you're in the hospital not doing well, and the goal would be to, you know, to get people out of the hospital and, you know, back on their feet. Whereas the vaccines would be given preventatively. So ideally, like, all of us healthy people would be given a vaccine, and then we wouldn't have to worry about getting COVID. So we're working to push all of those things as fast as we can. So that's what I'm working on.
Sounds really cool. Sounds like we can go back to campus soon.
Hopefully.
Okay. Yeah, I think that's pretty much it. Yeah, if anybody have any further questions. Yeah, and sorry for holding everybody for so long, like 10 minutes over the time.
If anyone has questions that they want to ask later or they think about later, someone can provide my email. I'm happy to answer questions about grad school or about this research. So feel free to email me.
I had a question about your opinion on undergrad research.
Yeah.
So a lot of undergrads aren't able to go to lab or go to school right now. What would be your strategy for finding a lab as an undergrad after COVID starts to alleviate?
Yeah, I don't have good suggestions for while everyone's home, because that's tough if your research isn't computational, which most of ours isn't. As far as once you come back, so I went to UMass Amherst for undergrad, which is not quite as big as UW, but it's still a large research institution. And the way that I got into research was I figured out kind of general topics I was interested in. And then I found professors who had research that matched that. And then I sent out a lot of emails. If you email one professor, it's unlikely that that person will have space. You might be lucky, but you have no idea. So I would say send out a lot of emails. Don't be afraid to follow up on those emails if they don't respond. And show that you're genuinely interested. You know, say, I looked at your website, or I tried to read this paper. The reason I'm interested in your lab is this, and I'm a junior, so I could work in your lab for a year and a half, or, you know, whatever your situation is. Kind of let them know why you're interested in, and show that you're interested. And a lot of people want undergrads to help and to teach undergrads, you know, why we love what we do. But yeah, I emailed a lot of professors and only one got back to me. So don't be discouraged if that is also the case for you.
All right, I guess this is time. Okay, so thank you everybody for coming today, and thank you Lexi, Dr. Walls, to come in here to review our work today. All right, I guess, yeah, I'm not good at ending, so.
Thank you
No problem. Thanks.
Extra. Thank you, Dr. Walls.
Thank you, and feel free to email me if you have further questions. Good
Yeah,
luck.
that'd be great. Thank you so
Thank you.
much. Stay safe.
Thank you. Good luck to you. You're doing really important stuff. So
Yeah.
Thank you.
goodbye, everyone.
Goodbye,
Bye.
everyone.
Real quick, before people head out,
Ends.
Oh,
does anyone want to participate in the next journal club? That's probably how we should end future